Thank you very much for having me here. And I'm really enjoying seeing those familiar faces and spending time with you again. And also like online, I just saw that very familiar faces there as well. So yeah, really nice to share something about the Unfold Toolbox today to you, because Unfold Toolbox basically changed my PhD from I don't know what's happening to, oh, that's cool. I'm really happy that this toolbox exists, and I'm really happy to share this with you today. And before I start, to go deeper, I actually uploaded the data and scripts and the slides already to this tab cloud thingy, and I'm not sure if I think I can just put it in the chat as well, right?
Mm-hmm. Sure.
Okay. Let's see. Nope. Oh, just a minute, I guess. Oh, interesting. Okay. So once you're there, the password is TwinBrain Workshop 4.0, and this without any spaces, but meeting group chat. Okay, the link is there now.
Yeah, you got it.
Nice. So for the people here, are you in the meeting as well, actually? No. Is there a way to share it amongst on-site participants?
Workshop 4.0 is the password, right?
TwinBrain Workshop 4.0.
I just...
Also
No, capitalized.
It's in the chat.
Okay. And let's go back here. Yeah, so you can just download during the talk and then be ready once we get to the hands-on parts. I also wanted to start with some historical facts about TwinBrain Workshop 1.0, and just to show you where we are when we look at the data. So once we record it, then we import it, then we prepare like our EEG data. Maybe I should use this laser pointer here. That should work for all. And going on, and then there's some nice analysis approaches which like are way broader than what we presented here. And we already had like an event unfolding workshop by Benedikt Ehinger and Olaf Dimigen themselves. So that was pretty cool, but it was I don't understand the question by Mona. Maybe someone can answer.
So
Okay. And but it was not recorded. So it's nice to have just another version of it, even though shorter, and me just as a user, not as the inventor or developer of the toolbox. But I hope that I can guide you through this still. And so I just wanted to point out that we are pretty at the end of the processing pipeline. So after pre-processing, like everything, getting it ready for an actual analysis, now we are employing an actual analysis, still not yet the statistics. So if you compare this like to other psychological data analyses, all those intermediate steps are just pretty not existent. You record data and then you put your statistics on the data. But like with EEG or mobile brain body imaging data, you have like tons of other steps to do. And I just wanted to show this again to create the whole scope. And so is this working? Nope. Weird. Yeah. Okay. Before I give an overview what we're going to do right now, I prepared a little Menti and I ask you to open menti.com, either using this QR code or the website and this numbers here. And I just want to go back. I'm not sure if you're familiar with Menti. If not, it's just an online survey, so we can just have some interaction with the online people as well. And just to get so I present this. Right, it's still here. I think I have to put it on. Everyone logged in already?
Yep.
Alrighty. Okay. Yeah, I think that's the way to go. It is still shared, right? This one? Yeah. Okay. Yeah, I just was wondering what your expectations are for this course, for this workshop. And so I put up some questions and just to gather this info. Yeah, this is already what I showed you. And so the first question, I think you were able to already answer some. At least there's already data. So maybe you can just give me a sign if you answered already. Yeah, most of it. Okay. So if you look at this, you all are really eager to learn. That's cool. And to discuss. And yeah, and money was not the issue. So it's okay. Now, I really like that with the TwinBrain project, that so many things, like really high quality things, are available for everyone. That's cool. And I think we're going to have some fun. All right. Next question was more open. What do you already know about this subject? Not much. That's interesting. That's exciting. So maybe you always have to interrupt and ask because as Urs already said, workshops are usually more advanced. So if you don't know much yet, so maybe sometimes we're going to be just a little bit quick. I know that EEG is cool, yeah, but it is also really a thing just hard to wrap yourself around it and to understand. I think after some years now, I still have no clue what's happening there. And so the toolbox is probably unknown, but at least some basic knowledge. Okay, thank you for putting your answers here. And the next question is just for now, chat a little bit with your neighbor. What's your main question for today that you brought, and what you really want to have answered. So, and for those online, just put this in the chat. So we just gather a little bit, that helps me to know what I want to target at. And yeah, I will also let you know, to be realistic, what we can do in one day and what we can't. So yeah, either you chat with your neighbor because you're here, or you put it in the chat so we can just, I'm just gonna share the chat for you. People are leaving. Okay. Sad. I can't read it then. Yeah, just for 2 minutes. All right. And does anyone want to say a question? No. Okay.
I think we have like lots of discussions already like beforehand. But yeah, I guess Nina, you can say something about your ideas, and then we can circulate here. I'm not sure if we're getting this insight, but
maybe I can sum up the main point.
Right. So I think here the intention to learn about Unfold toolbox, because I haven't even checked the website. Or I checked it like 1 minute ago, but I wanted to be a little bit surprised about what it does. I'm especially interested what's the difference between this approach, so this method, compared to other methods that kind of also deal with decomposition of data, like ICA or Amica. They also do like in this higher level picture, the same thing. So I'm interested what's the difference here.
Okay. So the question is, what's the difference from Unfold to other deconvolution approaches? Thank you. Anyone else?
perhaps like if they check the lecture we had from the first workshop from those two guys who basically built the Unfold, is that like enough information they'll have like to continue on working with your data, or it's even enough what you will be presenting in a bit?
Well, yeah, I have no memory about the workshop by Benedict, but I guess it was awesome because the paper is, I think, pretty much reading the paper, explain all the maths behind it, like even more than what I can grasp, but enough for me to understand like what's the idea behind it. And so I guess it is good for starting, but maybe it's also nice because I am applied now to several data sets, like to see like all the hiccups that I had and to give you the tips that I found useful. Sure. Yeah. Okay. Then I leave Menti here. Thanks for your participation. That took a little bit more time than I thought. That's okay. It's my day, so I can just rearrange the schedule. Yeah. Okay. Yeah, what I want to tell you now in this more theoretical part, we're gonna just like dive in a second, is like, yeah, how does that look like? What is regression-based analysis? And we were gonna look at this example. We're gonna discuss what is deconvolution, what are the prerequisites and the assumptions, and yeah, what's the basic analysis approach that the toolbox is providing. And then we're gonna have long sessions about like how can we actually do this, apply it to the data recorded here, and the noon and afternoon. Okay, a little bit more introduction why those things are actually necessary. And one application field that also like Benedict is pointing out in his paper is like MOBI research. So I brought you some movies, and I hope they start, from data that was collected in the B-Mobile. And yeah, it's lagging a little bit. But so we have different setups where tons of things are happening. People are walking around, they see a lot of things, they hear noises, and then we also have here Masha, but she doesn't start walking yet. In a similar setup, but I think the internet is overwhelmed. So this is Masha walking up and down in the lab in Berlin. And but what I basically want to tell you here is like, we have different processes at the same time, and that are all interacting with the brain, or the brain is interacting with the environment and everything that's happening. We see stuff, we hear stuff, we have like movement information, like from turns, from walking. We do have some responses to do, or have some other tactile input for our brain. And then we also like have from the environment, like from where we are, like especially in this data set, which is from my PhD, like we have other things around us that's happening. And to understand what of all those input measures actually is related to what brain response, the Unfold toolbox comes in very handy. So I'm going to show you later also example data from this outside the lab experiment, because this is where I was stuck. I had nice earpiece, Flink related, but I had like this contribution of the walking inside of them. And I was not able to apply any filter because walking is low frequent. And so I would like diminish all my earpiece as well. So I could not, like from my previous knowledge, I could not do anything before. And then I discovered that Benedict just published this paper together with Olaf. And this is also like the disclaimer. It's an awesome toolbox, and it's nothing I contributed to. I'm just using it. And all the fame belongs to those two people that did a great work. And it's also like increasingly cited. And I'm working on to promote it a little more because I think it's worth using in basically nearly every setup. It's useful. And I'll follow the paper that they published, which is in PeerJ, up here a little bit, and also take their example to explain the idea behind it. And I really recommend to read this paper if you're using the toolbox, but I'm gonna give you like a summary today. So I just gonna jump into their example. So it's face versus house discrimination. And so participants are just sitting there and they see certain stimuli. So there's a fixation cross, then there's a house popping up. They have to press the left button. Next stimuli is the face, so right button press. And what happens here is that we record this black data up here. Like this is like the EEG recording that we have in a seated, very clean setup. Like it is not true in the real world, but like it's just to show how it can look like this is like already you see there is like visual evoked potential in there, even on a event single trial basis. But there are different things happening at the same time and contributing to this black signal up here. So we do have a response to the house stimulus. We do have a brain response to the button press. We have a different response maybe to the face. We have, again, a brain response to the button, and so on. So, and those responses from the brain overlap, and they all contribute to the signal up here. And if we just average them, we will not see like the different contributions and and there are different effects playing into this. And for example, the first one is the difference that the reaction time actually is larger when we have to discriminate houses compared to faces, because we are experts in discriminating faces. And we need this like very early in our life. And so we are more experts in discriminating faces. So we are quicker. And this effect contributes to the data in this way that the responses are earlier, the minor responses, when we see faces, but it is not related to the processing itself in the brain. So we need to control for that. We have another fact or effect that's coming into play, and this is that different luminance of stimuli affects our brain responses. And we just assume for this example that all the houses were darker than the faces. So we have another systematic difference in the data, which is not caused by house versus face in the sense of their cognitive understanding. It's more about the luminance of the stimuli. And this looks then like this. So we have like a stimulus onset. We have a luminance effect on the P1, like the first positive response of the brain in our event-related potential. And then we have the button press pulling up. And now we see the contribution to all those different effects in the last row. So yeah, I actually did not explain what the event-related potential is. So if that's a question by anyone, I should start with that actually. But I see no questions here. So if online, this is a question. So at time point 0, this is like basically our event that we average our reactions to. And if we do not control for all those mentioned effects up here, this can be our ERP. And our event-related potential shows like a slightly difference in the P1, in the N1, and in the late positivity. But is that for real? This is what we want to check here. And so we also see that if we control for the covariate of the response time, so the earlier response compared to the later response, this actually cleans up like the effect in the late positivity. And if we control for the luminance effect, this cleans up like the effect we thought we have in the P1. So the remaining effect is only the N170. Like this is all like a clean example, not real data. And then we have the button press related contribution, which is also cleaned up. So this is like the actual effect. But because of all the overlapping and the different contributions of the other effects that are covariant, we suspect first that there are more effects than this, but the true effect is only this one, which is related to face detection. Okay. Are there questions to this example? Nope. Times says we have a break now. We can do the break still now and just go on with deconvolution later, or we go now for another 15 minutes.
I think we should finish with it.
Okay.
So we have like one conceptual.
Yeah. All right. On-site people decided we keep going. Okay. So we just go on more. So the idea behind this is deconvolution, like to understand what are the contributions of different underlying contributors to the signal. I already explained to you that this is like our signal that we record. We do have the different time points of the events in the ongoing continuous signal. And we want to get to this, which is the single contribution of different processes. And how we can do this, we employ or deconvolution, which is also known like in picture analysis. For example, I took here like this example of this dog bird picture, and now we want to understand, like for example, AI wants to understand what's on this picture, and then you see like, okay, there is some pixels that are dogs, some pixels are birds, and some pixels are different. So we see that this is like a convoluted picture. It's not clear. It's not a bird. It's not a dog. So different things add up to like an overall impression. And what we want to see is what contributes. So you can't read this. This is like, I think orange was, or green was bird. Violet is the dog. And then the pinky part is the human. And once we know what are like the major contributors, we can go the other step of deconvolution to see like how this information is present amongst all the pixels, and this looks then like that. So we have segmented the dog pixels from the bird pixels and the human pixels down here, and now we have them overlaid over the area of the whole picture. And we can see like how like the ratio of dog pixels compared to all pixels and describe this ratio. And this is basically also what the deconvolution in the Unfold toolbox does. Like it tries to describe the amount of brain activity that's contributed by a certain known process, which is defined by those segments, like which is for us the events classes that we want to look at. So for deconvolving, we have certain prerequisites to have in our data. For example, because it's a regression-based analysis, we need varying temporal overlap of the events. This means that the stimulus response time, for example, should not be constantly the same. So for stimulus response, this usually works because the brain is not like very, very strict. So we usually have there this time variance. And the other thing is what we can also like implement in our data sets is that we want to have a varying temporal sequence. This also helps. So that the interstimulus interval is not always the same. I'm gonna explain later because why those prerequisites are helpful for the regression. We do have several assumptions for the unfolding. And this is not moving forward. Yeah. So first of all, this first assumption is the same in traditional event-related potential analysis. So the brain response is the same for all events of one type. So this is an assumption we have, that the brain is always responding the same way. This is like also for event-related potentials, we average just across all the stimuli and expect that the remaining is specific to this event. So it's the same assumption, and it's still kept for the unfolding, even though there might be effects of priming or like adaptation to a stimulus, or there's also like some attention-related effects that those get ignored because like we still expect that the brain always responds the same way. I just want to point out this because there might be effects on time on task and stuff that we are still not considering of, but it is possible if you're interested in this to also include this in the unfold. The second assumption is that the variation of the interval between the events of different types does not influence the brain response to both of them. So the assumption is that if I see a stimulus and I'm super quick to respond, my brain does process the stimulus the same way, even though I have a stimulus and then I have all the time I want and then respond. And this is also an assumption that is questionable because usually I at least know of data that if you have to respond as quickly as possible because the stimulus is disappearing after 200 milliseconds or something, then the event-related potentials are shorter or earlier in latency compared to when people have just the time they want to until they respond. So, but still, this is an assumption underlying in the Unfold toolbox that this variation does not change how the brain prepares the motor movement for the response, and also like how it processes the stimulus. The third assumption is that the electrical fields generated by the brain add up linearly. So this is also like why it is regression based. So or linear regression based. So we expect that like the brain response to the motor behavior by adds on top of the processing of the stimulus. And this is something that's additional, like adding up and not like multiplying or something. How is this now solved in the toolbox? I try to go slowly through this figure and just interrupt me if you cannot follow, because I think it's good to understand. It's not necessary, but it's good to understand how this works. So now we have here the black signal again. So this is our EEG that we record, and time is plotted from top to bottom. And we do have different events happening. So for example, there is this orange event, there's a blue event, and then there is another orange event later. And every dot here is one sample. So we're really, really looking into like each sample of the data. And now we do, we code the contribution of every event in a matrix like this. So we define a local time around every event, which is here now 5 samples. This is defined by us. We also can use more, but we for now take 5 samples after each event for our matrix to code the contribution of this event. And for the first sample where the event happens at local time 1, there is an event coding of 1, and all the others are zeros. For the following sample, there is, this is like one sample later than the orange one. So this one goes through all the matrix until the matrix ended, because we defined it as only 5 samples. And at local time point 5, there is the next orange event happening. So the event coding has another 1 at the first position. Now we don't only have like orange events, we also have a blue event. And so we have the same coding for the blue events. So we have, again, like our defined local time of 5. And this blue event happens at the second sample. So here it starts, and then with every sample, it sample time passes, and it goes later. So this is like our design matrix that we create for all the events that we want to look at. And when we just look at one of those, we see like the contribution to this middle one is one weight for the orange, another weight for the orange, and 1 weight for the blue. And we can just write it down like this. And then there is a certain arrow, and I'm just gonna make the full figure now. So we have this design matrix, and we multiply it by those weights. And those weights are actually the things like our target measure that we want to know. We want to know how is this weight of this event And so it's an inverse problem. So we want to get to the weights. And this is what we also want to later on plot for our ERPs, regression-based ERPs. And we want to see like the contribution of this two events separate from each other. And of course, there's some error in that we have to account for. This is like basically the core math in the toolbox. And yeah, if there are no questions, there's a question.
Is this multivariate? So are all channels or taken into account, or do you pick the channels, or is it just one time series?
No, like this example is for one time series,
Yeah.
but the toolbox does it for all channels at the same time.
Mm.
So, but
So so every event is taken to be at the same time in the brain, regardless of which entity.
yes, yes, it's another assumption. Actually, I should list it. That it is not like temporal distribution of an effect that starts here and goes like to certain areas, for example, is not taken into account.
Yeah. And the 5 samples is like the default, because it also should depend on the sampling frequency, I guess.
This is basically what we later will define as an epoch. Like for our epochs, we know from traditional ERPs, we go like from -200 to 1000 milliseconds, for example. So we have, depending on the sampling rate, certain samples. And this is like basically tau. This is like only for here, just for the figure and to not have to like the largest matrix ever to understand like the basic. But yeah, this is defined during the process. Mm. Yeah, and regards to first question, you hand in all the channels, and at the end also can look at certain channels. And I think that's also what usually is done, that you just look at those channels of interest, but on the deconvolved signal. But it also works for example, for ICA activation. It's also a continuous signal. You can run the unfold toolbox on independent component activity if you're only interested in a certain cluster. So I haven't done this yet, but it's possible. Just need a continuous time series. Right. So this is the analysis steps that we need to take. So first, we need to specify the model, like which events with which effects we want to look at. Then we generate this design matrix based on those events of our choice. We time expand this design matrix. That means that we do this like with the local tau, like with local time points, like with the whole sampling, like we don't only have this first part for all events above each other, but we have it like for the whole, for each event, we have the whole matrix. That means time expanding. And then we extract those weight values, the beta values, what is the contribution of this event related activity to the signal. And then we can plot this and have some statistics. So it's majorly, it's 4 steps. And there are some more options in between that we can discuss more or less. And this leads us for a single subject, for example, to this result. Here, I just present you some blink-related potential from my data. So we have the intercept of a blink. It has like a nice potential component series of P1 and 1. And this is, I think, no, I actually don't know. I didn't plot which electrode it is, sorry. And I am not sure which is it. Okay, then we do have different times in the task where blinks happen. So there's ones where they just walk straight, where they turn, or oh no, they have the straight instruction, they have the turn instruction, or they walk straight. This was my experiment. Doesn't matter, but we just have different blink related potentials that change or that have a contribution on top of the intercept. So this is on top of the intercept. So when there is this, this is like an effect coding, but we can also during plotting define this, how we want this to be plotted. And here, this is my favorite one. You see the contribution of the step. We see like gate related activity in our signal. And this is like also not small. And this is now taken account for because we have this intercept around all the events about the gate. And then I added a saccade. And you also see here just the intercept of the saccade, like with a nice component array again. And as I said in the beginning, this had like a major impact on my data analysis. So on the left-hand side, you see like the data, how I went to the MOBI conference in 2018 and said like, well, maybe there is something, but it's overlaid by this weird thing, which I couldn't understand at this time. And everyone was just passing by saying like, oh, looks not good. And I was like, okay, that's sad. I really was looking for some input, like to help me, like how can I treat this? Like, of course, it was a small sample as well, but like for me, like the one group, like the Pro group, they were blinking with their gates. They just aligned their blinking with the gate. So I had a huge gate-related impact there. The red group didn't do this, so I even had like the systematic error. And after using the Unfold toolbox, this was really cleaned up and nice and publishable. And so, yeah, this was just the moment where I became a fan. And the next step would be an unmixed, like a group level analysis, because Unfold Toolbox doesn't do this. And there is this unmixed toolbox. However, it's abundant. So Benedict is no longer working on it. And he says, I don't recommend it. It doesn't work. So for the group level, there is no solution yet. But he's working on a Julia version of Unfold. I don't speak Julia, so I didn't check it out yet, but he says that the Julia version of Unfold already allows for like large linear mixed models. And this would be kind of the solution for a group level analysis. And this would be the way to go for, but I haven't checked it yet. What I usually do is I export the beta values and just plot them nicely on a group level.
Can we go back like to slide? Just
two
like
slides.
A very basic question on this one. So here, what you want to say, you're showing on the left side FZ and CZ electrode. And then you said on the right side you have the FCZ, so it's another electrode. Was it like over there on the left side all the gate-related artifacts? And then with the deconvoluted option,
Yeah, basically, yes. I did not have the data saved, like the bad data. So I could not recover like the FCZ, or I can do this, but I didn't take the time to actually recover this. And I just took, that's why it's different electrodes, but it's the same area. But all the electrodes look similar because like this gate related impact is pretty big, especially in this frontocentral area. Because I think actually it's like the movement of the cap due to the heel strike. And it's not really gate-related processing of the brain, but this is my personal opinion. And it definitely was excluded after, like you see this immense contribution that just really adds on top in this time area of interest, which is difficult. Mm.
So then the time zero is like the eye blink here. Say.
Here it is the eye blink, yes. Overall, and here it is the saccade, and here's the step event.
Mm-hmm.
So, but for those four, it's the eye blink.
Okay. Because then for the second then slide, you could basically have the comparison of like standing versus walking, not having those trajectories and then trying to make
you could have, but I didn't have the data. And you also don't need it anymore then, because processing may be different in different motor states. So,
sure.
So you're also not sure if now the motor state changed or is it just a clearer signal.
That looks nice.
Yeah. Yeah. Okay. And just for Nina's question, regards what does ICA do and what does Unfold do? ICA is a spatial filter of where the source is located in the brain and this contribution to the skull EEG signal. And Unfold is the same approach, but on a temporal way, like you have a temporal filter. On like the contribution of each of the different events to the signal.
Yeah. Did you try using ICA in this data?
Yeah. But I never did like an intensive clustering and looking if I can remove all the gate-related artifact like that. Mm-hmm. But it would be nice to compare that actually. If you want to dig deeper into the all right. Okay, this was basically the theoretical part. I'm not sure I didn't see any pop-up questions in the chat. There were any questions? No questions. Okay, so I would proceed into a break. Do we still want to take like 15 minutes?
Yes.
Or 20? We want to start 10:30. It's 20 minutes. Yeah. Okay. So just make sure in the break that you download everything related to the data and the scripts, and then we can go on. Hello.
Hello.
Welcome back. So just for a slight start, I'm gonna open another Menti. Where is it? Okay. So you already can open the website because I don't have a QR code right now. Maybe I should leave at Rome and enter the house web Wi-Fi. Taking so long. Send.
Yeah.
Okay. Maybe you cannot read it. It's 3196409. It's the code up here.
Mm-hmm.
And most important question first. No, it just was there. 3196409. Oh, okay. You need to choose puppy or kitten. Forced choice. There is no both.
There's only one choice.
So maybe 6, 7, 8. Okay. How many people are online? Checking if everyone is following. 10 people are online, so okay, true. Well then 8 is probably good. Okay, so you can go to the next question, I think.
You fix that video.
Sorry?
Do you need to extend the video? Duplicate.
No, it's fine. I just want to know what EEG data analysis tools you know and use mainly, and how this correlates with your preference for kittens or puppies. Just because I can. Okay. So there is some EEG knowledge, some field trip, nice. Brain vision analyzer, no. So Torben will have some work with us to do. But it's only 7. There's the 8. Okay. So I think, what do we learn from this? No causality, of course. Okay, thank you. I just wanted to know where we are together. That's already it. And we proceed for our first hands-on part. And what do we want to do in this first session? It's gonna re-explain you the task, which Urosh already introduced. And then we gonna create the necessary event structure, go step by step through the Unfold toolbox, and hopefully get to the point where we plot some better values. We'll see. For the task description, we do have a 2x2x2 mixed design. So we have this person standing on the force plate and wearing an EEG headset and looking at the screen where some stimuli appear. Either they're stroop, like incoherent or coherent. And so we have this within subject factor of task, if they just have a comfortable standing or the semi-tandem stand. We have a silent count task, so they have to count a certain kind of stimuli. So they attend to some of the stimuli and ignore others. And then there's a between subject factor of group, if they are healthy or have a early starting Parkinson's disease. How did this look like? So there are different 3 minutes blocks, either single task, semi-tandem stance, then single task with the stroop. Single task refers to the motor task in this way, and then there is the dual task semi-stance. Is that correct? Yeah. And this is how one trial looked like. So they have a fixation cross for 200 milliseconds, a stimulus for 300 milliseconds, then a black screen of 500 milliseconds, and then the new trial with the fixation cross started. And there are some outcome measures of interest. So behavioral, we want to know whether, like, how the center of pressure variated across the task, and how well they counted the to be attended stimuli, and then the neurophysiological measures from the continuous recording to stimulus locked event-related potentials. And this is basically where we focus on in this session. And now we're gonna already proceed to MATLAB. Oh, here I had the Menti, but we already did this. So I know that some puppy people know EEG Lab. We do have some requirements, so we can start with this for now. Everyone needs a MATLAB license. Everyone needs EEG Lab. I'm currently using the 2022.0 version. Doesn't mean it has to be this one, but yeah, I just put in the link to download this if you don't have EEG Lab yet. I can also show you how to set the path for that. IC label is already part of EEG Lab, so we just need to upgrade to the current version. And then we need the Unfold toolbox, which is not yet. The links. Yeah. Yeah, but also everyone can download the slides now. So they already have the slides, if that's easier, but I can also try here.
I can do it if it's easier.
Yeah, that would be nice. Yeah, because I always have the presenter mode here and no copy pasting. So just give me some feedback where you are, if you're all set, well, what you need, where you need some guidance. So for the Unfold toolbox, you just open the GitHub page and then either you use the link, I hope that works, or you clone the main of this GitHub.
Place it in the EEG toolbox folder. Yeah,
it doesn't have to be there.
Okay.
It is more tidy if it's there, but you just need to set the path that it's that MATLAB finds it. Of course, you have to unzip it and extract the files somewhere.
And the label 1.4.
Yeah, the current version. I can also show how to do this, but yeah, I think I quit sharing here. Can I? Does the link work for the Unfold? Not for you? Okay. So you basically just open the Unfold GitHub, and then if you're in the main page, you can just here for code and then clone Yeah, maybe I should switch now to. Okay. Yep. What did I do? Why is it not sending anything anymore? I kind of stopped the contact tuning. But how? That's the question. How did I do this? Show now. Replicate. I kind of disconnected to the second screen. Wait, something's happened. Yeah, works. Oh, sorry. All good. Okay. I just needed it to be duplicated right now so I can see MATLAB. You can see MATLAB? Okay.
You can, but the online cannot.
Yeah. I need to share it still, but.
You share the entire screen.
Yeah. Share screen. Share. All right. Now everyone should see everything.
Yeah. Yes.
Okay. Did cloning the unfold work? Everyone unzipped and put it somewhere. Okay, once you have everything downloaded, you just make sure that in your home of your MATLAB, you have set path to eglab and the unfold master.
Mhm.
The unfold minus master, it should be in the unfold folder, but maybe it also is sufficient to use the unfold folder.
Right.
It's me.
Okay. All right, give me a thumbs up if you're done. It's not everyone yet. Where you see. So if you have MATLAB and the path is set, you can just open the 3 scripts I shared with you and that you downloaded in your editor. Maybe your layout does look different, so you can choose your layout here. I will always like the default option. So you have the editor incorporated into the MATLAB workspace.
You have a script PDICA something.
Yeah, no, that sounds, this is 3_ICA interpret event underscore PD for you. I just didn't rename mine, but I renamed it online because I thought maybe it doesn't make sense for you to have the numbers. But for me, basically, 1 is always importing, 2 is pre-processing, 3 is event creation, 4 is unfolding. So that's just the way I do it. Sorry, you have to cope with that. But that's why there are numbers. Go. We definitely will start with the 3_ICA interpretation event. This is basically the last workshop applied to this data set. And I added some lines.
Did we change the paths to our data?
Yeah. But not for this one. This is just, I want to explain, but you need it here for line 33. You should enter your path to the data.
Yeah, already in line 9 it starts.
Yeah, no, it's not that necessary up there. This is just something I wanted to show you before. Here it is important.
Which one again?
33.
Okay.
33. Adapt this path according to your path. Internet is unstable. I think I switched to the other Wi-Fi.
Yeah, but luego del tema del uso proban más luego tú estás ya okay conmigo
Something more that makes sense to me. Okay. That's la data comune se deve non so che ma non so.
Okay, hopefully the internet is now better. Okay.
Just explain to the others. On 33, you have like the name of your folder is TwinBrain Workshop 4 without data. I think.
Yeah. You should go until your downloaded folder.
Yeah.
Okay. So, okay. I think nearly everyone is there. So this is also just something that's still there. It's probably not needed, but I just ran those lines because I did not want to mess up my code. So you can simply either run the section using this one or mark those 3 lines and then press F9. So we then see that this is like stored in our workspace somewhere. And what I just wanted to show you here is that I used the data that Mansa shared with me and concatenated it again. So I had all the Amica Z files, concatenated them, and merged them and saved this as now called ICA preprocessed, or no, preprocessed and ICA. which is basically just the data that Mansa sent me. So it's pre-processed already. I didn't put the pre-processing steps here, but I just used yours the way it was. And when I checked it, there were quite some immense artifacts in there. So we'll see. We can just go for it for now, just like it is. And then if everything's messed up, I just put in here the timings where I thought like maybe it makes sense to skip them. But yeah, for those at least 2 people that are joining that have never analyzed EEG, we can just type in EEG lab. Yeah.
For the PET segment data, is it the seconds that you are?
This is seconds, yeah.
Brackets?
Yeah, but
These are the times to be excluded.
basically this needs to be no time.
Yeah.
Because otherwise it will only keep them. But you don't need to run this line.
Yeah.
I just wanted to show what I did. There's some warning. Okay. So MATLAB is there, I guess. EEG lab. Where is it? I always recommend you to check your data, to look at it, to see it, to get a feeling how EEG data looks like and what issues are there. So I just use the GUI for now to load the same data set.
Which one?
The one provided. Yeah. Yeah. That's the only one that has the standing data. So, and then you can, if it's loaded, you see like some details about the data set here, like the sampling rate, how long it is, what's the reference, if there is some ICA solution coming with it. And then I use plot and channel data scroll. And I think it's at least worth to look at the data first before we do any analysis with it. And due to the many channels, it's like intense data. So it's even, and once we have the plotting, we can change how many seconds we want to see and how many channels. I would reduce it for visibility reasons. Just go for 32. No, 64. Okay. Then it's still a little messy, so I go higher in the Y scale. Now we see 64 channels for 25 seconds, and we already see here our events of interest. So we have 555 as the fixation cross. Then we have 1002 and 1001, depending on which stimulus we have there. We do see some nice blink potentials everywhere. Just blink movement. The data, and I just want to show you. Yeah, I just wanted to show you for now, like those very strong artifacts. Like it looked like even like the whole setup was like it crashed for a second or something. So and there are many cleaning options and stuff that works, but most of them rely on that neighboring channels behave differently. So they take into account if one channel is really messing up while others are stable, then I can remove this channel or interpolate different samples. But for something like this, where all the channels mess up at the same time, usually the algorithms do not check, okay, this is an artifact, because everything is the same again, even though it's artifact. So that's why I chose to check whether like selecting the data and reducing those time areas helps. But I think for now, we can still work with it. I just wanted to show it because sometimes those huge artifacts can mess up your analysis steps. Okay, that's our EEG data. Good. That's okay. So back on to the script. So this one we can ignore for now. This is for later for the dual task walking. And now you can run only like your line 33 because it's gonna use this path several times somewhere in the script. So if you run this like with F9, what did I? Oh yeah. But you need first to define your subject index, and it's not 3, it's 8, for example. Then I rerun this one. Okie dokie. So this first section is basically only for loading the data, which we did now via the GUI, and then plotting the data and look at the data. So you can run this if you want to check your data. So we just skip this now because we did it. Then I have another section where which I described more in the workshop, in event creation workshop in 2021. But I still wanted to keep it in here. So if you're interested in checking out how I came up with this blink detection way, we can go deeper in here, but I think I don't want to use too much time on this right now. So now we have the script part section where we automatically detect independent components to represent vertical and horizontal eye movements. Again, here we are now participant 8. And yeah, here your path subjects gets redefined. Either you change it again, or you just comment out this line. So it will just take what you already defined. So it loads the data, and this section also contains some automatic gate-related potential lines that I just kept in there for information. So because I use the same script lines for my data where I have an accelerometer in the EEG signal. So I just used this, and I thought like maybe it's nice for someone who has this data as well to look into this, but you can just ignore it for now. For the IICs, we define here a prototype for horizontal eye movement, and that we correlate to independent components that were labeled via IC label as highly I activity, and then we save this correlation info and assign the index and save it in the dataset. So basically what you can do is just go into this section and run it. It should run. Or otherwise, maybe your IC label is not there, or some paths are not set correctly. But just try this for now. If that runs, basically a lot of other things should run as well.
And so you skipped the previous section?
Yes.
You didn't run?
This one?
Yeah.
No, it's more like an explanatory. We can look into this if we have time.
Okay.
How I selected the blink peak detection. But it's also already in the other workshop, so people can look at watch it.
The line 204.
We are now in starting section 94.
And it runs until 204?
It runs, yeah.
Okay.
And it gives you two figures that are basically the correlation. And I think for participant 8, it's actually only one IC that takes both info. So if we now just want to see which IC was chosen, you can just mark your parameter and run it. Then it's 3. And it's also 3. But you can also, instead of doing this, you can also check your workspace where those parameters should be somewhere, index here. And then you see here it's both 3. And the other option is to click on this variable, double click, then another window section opens where this is written. And we did save this now into the EEG struct. So I'm not sure how familiar you are with the EEG Lab EEG struct. There's tons of information in there. And you can access that also via double click on the variable, or otherwise you can also type into the command window EEG. Then it also opens the struct. Getting a little convoluted here. But you basically have all the info. You have the data in there, the sampling rate, the times, the independent component activations, the spheres and weights, all connected to the independent component analysis. You have the different channel names, the reference, and the dipole fitting. And what we did now is to write like an EEG ETC. There is some info from different pre-processing steps, and also the ICs for events is now created because you ran the script section, and here's the info of that we just saved into it. This is handy because we later on other steps can just get to this information again without having to rerun. So this basic info about the EEG lab structure. And then we want to,
Sorry.
yeah.
How do you get this prototype H in your current development?
I think this is basically work Marius Klug did. So I think he just used a prototypical IC that we choose manually, and then saved the weights as the prototype. But I'm not sure. I have to ask Marius how he created this array, because that was his contribution.
So each element in the array is one weight of a component.
No, it's a spatial filter again. So it's one. I think it should have. Let me see. What's the size is? No. Yeah, it's every channel. But why it's a 67? This is maybe why it messes up sometimes. No, it's not a bug. It's like just a misalignment, because like the prototype is only for 67 channels, which means with 128 channels, those weights need to be adapted. But actually the correlation should be still similar, because I think with the 128 channel setup, you have first, like they overlay each other, but that's different for every system. So yeah, this is something we should point out for that it is like setup specific, which channel aligns to which measure here.
Okay, but
I
the
should-
element should correspond to a channel.
Mm-hmm. But it worked somehow. So I think it is like with the system you used, the first 64 channels still align with a 64 setup of brain products, for example. So
I think the data set has 128.
yeah. But I think it's going from the front to the back. And then the other 64 as well, again, going from the front to the back. So the correlation of the first 64 channels is, again, there for a weight matrix like that. I just noted it down here. I think that's important. Thank you for pointing this out. Okie-do. Then just let's go one section further. In this section, we now use our new file where we did select the ICs. And now the actual event creation happens. So we smooth the data, we do create like an EEG vector, some derivatives to get to like a saccade detection. Then we create those saccades. Then we create the blinks. And then we delete all saccades that overlay with blinks. And then we save this info, plot it, and all those out-commented lines here are about the gate. I still kept it in there. And then at the end, I remove all I components because I don't want to have this contribution in the unfolded data. Like even for the blink-related potentials, I don't want to have like the muscle movement of the eye in my data. I want just to have the brain response aligning with the blink and save this. So you can run this section that ends at 440. And let's see what we get here. Yeah. Sorry. Yeah. So we're still just preparing everything for unfold. We're still not there.
Sorry,
Yeah.
Did we create a new file with the IC selected?
Yes, that should be like the end of the other section. No? Here, like in line 202, it should save. Okay. So there, if it worked, you should have like this figure showing the independent components from 1 to 10 with the new events. And we chose independent component 3 for both of them, like for saccades and for blinks. And you can see like those really nice blink potential, which is detected. And you see that all my peak detection works on thresholds. For example, this one is probably a blink, but it did not reach the threshold. So it ended up being a saccade. And this is also something that I just want to be transparent about. It's not for sure, but most of the time it worked. So you can improve this if you want to. But for now, it kind of runs. And you also see that within this troop task, participants tend to not blink. So basically, you see, again, more blinks after. And also, you don't see a lot of saccades. This is also because, or this is one reason why the horizontal eye movement detection algorithm did not really work so well, because it's just a lot of looking straight and not too many saccades. But you also see that here with this artifact that I already pointed out to you, this is like detected as a saccade because all electrodes are affected. And for now, this is not too bad, because even if it's just detected as a saccade, it gets into the rubbish bin of saccade-related potentials. But if we want to look at saccade-related potentials, we should take care that those times are removed.
I'm sorry, so the blink detection was done by the peak finder, or?
Yeah. So it's all in the section we just ran.
Mm-hmm.
So for the blinks, this is for here, up here, that's for the horizontal eye movement. Yeah. So you have, I did like an adaptive approach with the find peaks function. That's what I used. I defined minimum peak prominence and minimum threshold based on the actual data from the independent component, but the others were like default values that I chose due to the nature of blinks, based on like EOG papers where that like, okay, that's a reasonable.
Parameters that you usually play around depending on how they blink. So
yeah. But I usually try to not play around with them. So I keep them for all. So I tried to set them once, like on my first data set, in a reasonable range. And I try to stick to it and to not adapt it individually. Or otherwise, like come up with an idea how to use like percentiles, like to have this automated so that another person can just rerun and get to the same results. Yeah, so genau. So here we create the eeg.event structure, and this one, but has still only the type, and we are still lacking for the unfold toolbox the naming. So what did I do here? I think the rest of the script is not important for now. Yeah, we did basically everything I wanted to do with this script. You see here, the following section is where I removed the artifacts. And here also you see, Mansa, that I used the right tag, no time. So yeah, that was the actual one that I ran. And now we switch to the other script, which is called unfold PD. That's the standing script. Okay, we're now in this for_unfold PD script.
And I guess we need some time
Yeah.
to load it, but I have it. So the next step is the unfold PD
Script. Yeah.
We are getting an error because there's like no some information on the ICA cannot get loaded. I think I myself was
Okay.
and we're wondering like we're gonna be starting this like run.
So did you check, can you check on your EEG lab, this one? The manage EEG lab extensions, just open this one. So it's file, manage EEG lab extensions.
So I guess it will solve. For me, it didn't work either. So I closed the EEG lab, and then EEG withdraw, and it loaded the reload.
Okay. Like one thing you can check if IC label here is bold. That's for sure has to be set.
Mm-hmm.
Yeah. Okay.
Let's wait a bit more.
You know what's our problem now? When we already know the PG file at line number 29, this one, so when we just have this subject, we don't get this merit of the ICA.
Okay. Yeah, we can recreate this.
I don't know how it looks for.
Can you just run this line again? No, this one.
you tell me which one?
Usually this recreates the ESC equation. And it's a really important line that you should always run every step you do.
But it can.
Because it checks like this, checks where I just do it for the online people as well. Like this line here, I just try to do it after every step I do because this just checks whether the EEG struct of EEG lab is correct still. Like if everything is aligned and it like one line of this function EEG check set also recreates this ICA activation parameter.
But it doesn't.
No, it doesn't.
I saw that the activation state in the EEG spatial filters.
Here. Can you try to type this? Is it large enough? Should I increase? Just don't really want to increase the line what's called the type size because otherwise we don't see anything anymore because of the size of the screen. Not to solve it, just to look if it's there.
Yeah, so, yeah, I mean, it's not. Then we haven't had EEG, but if it's some, spatial. But I need to get more of the.
Okay. I'm not sure if online people are actually following, but there are no questions in the chat.
You can also just continue and then.
Well, yeah, many things are kind of following. So, but then you basically cannot run the codes. But yeah.
There are more than the EEG, I see it might be there, and I want to.
Yes.
You can ask also if one is, I guess, she's following. Works compared to.
Yeah. Any questions online? Is it running for you? Nope, still not.
No, I have to get inside. Okay. Maybe you can just put line in there. Which one? The one that we create. Yeah. Which one?
But it's not the same. It's not the one. Because I thought that's the red and the blue, but it's different value for the text. It doesn't have enough what's inside of the pages. So because I don't know what's on it. Because the whole times here it's
Yeah, then let's do it differently. Where are you? Token.
Use
18 at this. But basically you need this line. Are you in the Zoom call as well? Mansa or Nina, are you in the Zoom call as well?
Yeah, they are.
Okay. Then I just put this in the chat, and can you try?
No, no, it's sending.
Oh, Mona, you want this? Everyone, where are you, everyone?
The first option.
Meeting group chat, okay. So this is like the line, this is like in the EG checkset function. Because I did have this issue as well, several times. Sometimes when I use this line out of this function, it solved it.
yeah, no.
Yeah, perfect. Yes, please. Okie dokie. So I can just go back. So can you run those two sections from the script?
Let's try. The subject again, 8, right?
Yes, now we're back on 8, but it should be already set on 8 because I created two different unfold scripts for the two tasks because they differ quite a lot. And so it helps to keep it clean. And here, I'm just waiting a little bit more that everyone is on the same page again. Yeah. Your path adaptation to your own path. What time is lunch?
No lunch.
No, I just want to get my timing straight.
We understood. Noon, so then.
Both? Okay. Good. Then we have sufficient time to go on. Hopefully.
That's getting errors.
Yes? Okay, what's your error?
So, yeah. So such pattern.
Oh, yeah, you need to remove from this underscore.
Here.
Yeah. Because I didn't create this review, we just keep the other sets for now. It's remove the no only.
It's such screen.
Yeah,
But they removed the screen or?
So this should remain EOG events and no IICs.
Okay, got set.
And this should be found. If this is not found, you have to rerun the last section in the other script. Where the IICs are removed. Oh, so you're running the full section?
Perfect.
Okay.
Just the section, so I guess the whole one.
Yeah, but it's okay. It's okay. Because it's good to know if it runs for you.
Let's see.
Because I struggled a little bit last days to get it run, and it's good to know if it's run somewhere else.
No, let's see.
Okay, where are you, Manzanina? Nice.
Yeah, Deluxe, yes, yeah, I got it.
No, that's
That's not? Okay.
so iteration 400 and the time function brand.
Okay, but at least the unfolded brand. Yeah, let's see. You go to the really unfolded.
Nine-fold.
The unfolded. And then publish.
This here.
Yeah, let me check what I did differently.
The magic line. Yeah.
Okay, unfold is not here.
Mhm.
Hmm.
Taking that
Yeah. Okay, where did I get this one from? Guess you need this one. Nope.
The problem is whenever we load the new data set, we have to recalculate the ICA. I mean, the ICA. We are kind of.
Yeah, I think we need to get the CRUM toolbox as well, because it's used for the plotting. So you basically type in git CRUM, and then this was the first hit that was shown to me. And then you need to download this as well.
Yeah.
Okay, I can just put this in the Zoom link. Chat. Yeah. But once you're done with the once you're done with the creating the EOG events no ISC set, we can just look into the other script snippet before we go on with running stuff.
Okay, yeah, we created IC select. For which one?
No, once you've, like, this one is created, this one here, you just, like, it's the last thing that you should create based on the 3 script. So you only need to run the script until 440.
Okay, yeah.
We got it now.
You got it. Awesome. Perfect. Makes way more. No, let's do this later. We can just like now look into the script, and then we can install the CRUM to run it. We can also do this after lunch. Yeah, now we are in unfold_PD. And I just want to show you how the event structure has to be, because I now concatenated the data for all tasks. Then we created events for the blinks and the saccades. This is what we load here. Then we remove the eye components to reduce the impact of the eye movement on the EEG data. And now we have to create the structure. So we're now in line 17, where I start to create the events as we need it. And what I first did was I again searched for the events that show me which task part I am right now. So because I want to later on have in my model the tasks as a factor. So I need to know when was a blink within a single task, when was a blink in dual task. So I look now in my concatenated data, where was the start of a new section of the event. So I save this. We can just show it now for here. I save this in this task start latency variable, where I now see, okay, each task started at event index 9, the next one at event index 115, blah, 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 till the end. So those are the start events of each section. And now I make another loop through all events. And as long as the event is before the fourth entry here, which basically 4 is the switch from dual task to single task, because I use the dual task first and then the single task later. And so here, before 399 is the switch to dual single task. So all the events that have happened before get in their event struct, eeg.event struct, a task label, which is either dual task or single task. So then later on, I can use this label to use my factor and to do like a comparison of both tasks. And similar, it happens for more in-depth analysis, like we don't need to do this, but we can do this. And that's why I did it, because we can. I renamed all the 555s to fixation cross. I renamed the 1001s to stimulus. And which kind of stimulus? Oh, wait. This is not correct here. Why is it in here? That should not be here. Yeah. That's why I was like, I did actually create a...
have a tentatively born.
Yeah.
And this should already be coded within the 1001 and 1002. The 1 is attend, the 2 is ignore. So you don't have to run the slides here.
Yeah, yes and no. Okay, so I maybe just rename this. How do I call this? 1 is attend, right? Yeah. So. Woohoo!
Because I didn't see the reason why.
No, yeah, we switched.
Oh,
All good.
Where?
Okie dokie.
so the type to just put in stimulus. Right?
Yeah. I renamed them because I need an overall name. So the type always has to be like the overall event class kind of thing, like the event. And then the factor level that you are interested in should be like another level. I now named it attention, so I should rename it, but I did not include this yet in the model, so it can also just go. Sorry for the confusion in the script, so it looks way easier now.
Can you check?
Yeah, I just changed it now, but it's not totally wrong, but it's also not totally right what was there.
I think you just made like one script with both tasks. Is then making more.
Now I do have the dual task walking, which has more advanced renaming. So,
Can you tell us once again, like, what did you change?
yes, I can just put this in the chat. Or you cannot copy. Can you copy now? I think it.
Okay.
Are you in there yet? Okay. You.
34.
Yes.
Unfold PD.
Yeah, unfold PD.
34 until. So all those.
All the others can go. Yeah. And they are not wrong because those events just don't exist in this data set. So, but yeah, just for cleaning purposes, this is way better now. Okay. So, and now we do have like everything set for the event structure that we would need when we run this. So we have our main factors that we are interested in, and we have the factor levels in the below that, for example, like the attention or the task. For now, I only added the task into the model, so I kept it easy. So this is now when unfold starts. No, I don't run it. I just explain it first, and then we can run it later. Are you there?
So I see there's a file that's called artifact rejected as well.
Yeah, you have to remove this for now.
Parts of the data that are okay, right?
Yeah, for now we just use this one, which you already created.
Okay.
This one. You can just remove the artifact rejected part. Okay, so I just want to explain the model first, and then we can go for lunch.
Okay.
Waiting if everyone's there. Yep. Okay, so once you want to use the Unfold toolbox, you run and initiate the toolbox. Then we need to create this design variable struct where we define how long our epochs should be. This is of your choice. So I just used a longer pre-event because of the blinks. Then you enter all the different event classes, event types that you want to have in your model. And the order here is somehow important because it should align to the formula. So I here just included the stimulus, the blink, and the saccade. I did run a model also with the fixation cross, and we can also do this later just to see how things go and not go well. and then I design the model and I say, okay, for the stimulus, I'm interested in the intercept to see like the overall ERP regression based. And also I'm interested in the categorical factor of task, like is it single task or dual task? Similarly, I could also add here what we just did, the factor attention. And here I now say I want to see the interaction. If I want to see the main effects, I should just first have just the categorical factor and then the interaction. But as this is like way too big for a start, we just stick to the task. And for the blinks, I'm also interested if there's a task-related effect on the blink-related potential. And I just want to see if saccades contribute something. So they have just the intercept formula. So this always has to align, like then the order, those are up here, should align with the order of the formula for all the different event classes. Then we just hand over all the channels that are there. Yeah.
What was your intuition? Why did you choose such formula? Like, for example, for Blink, why would you add a test?
Because I expected less blinks when dual tasking compared to when single tasking, because single task here is the motor task. So they only have the motor task to do and no stroop. And what we already saw in the raw data was less blinking while stroop task. So it may contribute. You can also remove this. If it's not of interest, just delete it. Just keep the intercept so that you could request out the blink-related potential out of your stimuli processing. It's totally fine to do this. Like it's just a matter of interest. And then you create here the design matrix with this function. You time expand it. Then there is an inbuilt way of artifact detection with a threshold. You can define this. I think the default they recommend is 120, but I'm not sure actually. Would need to look this up. And so artifact loading samples will not be included in the regression model. Then you fit the model, you condense it regards to different factors, not factors, event classes, and then you already can start with the plotting. Everything that's below here is basically, already plotting the results. So once you had got everything prepared for the toolbox, it's not a lot of lines to solve. But you saw that we just took us an hour to prep the data in a way that we can run it. But with this, being able to run it, hopefully, I would just transfer into the, what is that? Opening. No, I don't want to open. You're already in the middle of it, to the lunch break. And then we can run afterwards. And I ask you to download the Gram toolbox.